This is Peter Voorhees. I am with Atrium Health, Levine Cancer Institute in Charlotte, North Carolina, and I'm faculty at the Wake Forest University School of Medicine. I'm at the 2025 annual ASCO meeting, where I'll be presenting data from the long-term follow-up of the CARTITUDE-1 patients treated with the BCMA-targeted CAR T-cell therapy, cilta-captogene autoleucel, or Cilta-cel, for patients with heavily pretreated relapsed refractory multiple myeloma. So many already know a lot about the CARTITUDE-1 study. So this was a study that enrolled patients who had triple class exposed disease. So what I mean by that is that they've been exposed to CD38 antibodies, they've been exposed to proteasome inhibitors and immunomodulatory drugs. The patients on the study had to have had at least 3 or more prior lines of therapy, or be refractory to both an immunomodulatory drug and a proteasome inhibitor, and have disease progression in their most recent line of therapy. Now, we already know that this patient population was heavily pretreated. The median prior lines of therapy was 6, and the majority of patients had not only triple class exposed disease, but they had triple class refractory disease, so disease that was resistant to CD38 antibodies, at least one proteasome inhibitor, and at least one immunomodulatory drug. With that context, when you look at a similar patient population that was followed in the registry study Locomotion, we would expect the median progression-free survival to be 4.6 months for this group of patients, and the median overall survival to be 12.4 months. The median progression-free survival from CARTITUDE-1 has previously been reported and published, and that is nearly 3 years in duration, which smashes the 4.6-month median progression-free survival that would have been expected, prior to the advent of Cilta-cel and other available therapies now in the space. With this long-term follow-up, so the CARTITUDE-1 patients have since transitioned to what we call the CARTINUE study, which is really to look at, for those patients responding, how durable are these responses? What does the overall survival look like? And are there any additional safety signals with longer-term follow-up of which we need to be aware? So with that context, what I can share with you is that 5 years after a single infusion of Cilta-cel, a third of the patients that were treated with Cilta-cel are still alive and free from progression of their multiple myeloma. I can also share with you that the median overall survival is just over 5 years. Again, comparing to the expected 12.4 months median overall survival, really an incredible result for this group of patients. The CARTINUE study will actually follow patients over a 15-year period. And again, we'll be looking at survival, for those patients. Again, we have a third of the patients that have yet to progress. What are things going to look like over the next 5 years for that group of patients? What is it going to look like over the following 5 years? Are some of these patients, for all intents and purposes, cured? I think, we have to be a bit cautious how we use that term, but I think there's absolutely no question that this is unprecedented durability of remission that we're seeing here with this therapy. CARTITUDE-1 was the study that led to the FDA approval for more heavily pretreated patients who've had 4 or more prior lines of therapy. The CARTITUDE-4 was a subsequent phase 3 study that has since been published, and it's been presented at several venues, which looked at Cilta-cel versus standard of care therapy for myeloma in patients with relapsed myeloma who had had 1 to 3 prior lines of therapy. And what that study has established is that progression-free survival is superior for those that receive Cilta-cel. And not only that, overall survival is improved as well. So now the approval for Cilta-cel is for patients who've had 1 or more prior lines of therapy. Why that's important is one of the interesting things from a correlative analysis perspective from CARTITUDE-1 is that patients who received a more immune-fit CAR T-cell product appeared to be more likely to respond longer. So when you look at these patients who are free of progression and alive 5 years or more after their Cilta-cel infusion, there is a higher proportion of naive T cells in the CAR T-cell product prior to infusion. And at the time of peak expansion, post-CAR T-cell infusion, there was a higher effector to target ratio for those long-term remitters, and there was a higher proportion of CAR T cells with a central memory phenotype for those long-term remitters. So we think, that as we apply Cilta-cel in early relapse, as was done in the CARTITUDE-4 study, we should see more patients who have an immune fit CAR T cell product. And so we're hopeful that there'll be an even higher proportion of patients who achieve long-term complete responses. And we now have the ongoing CARTITUDE-5 and 6 studies that are looking at Cilta-cel as part of frontline therapy. And we're hopeful, that the fitness of the CAR T cell product there is even better. And there we've got very effective strategies in the front line, where we can control the disease burden and optimize the effector to target ratio going into the Cilta-cel infusion. So we're really excited to see what the durability of responses is going to be in that group of patients. So in the CARTITUDE-5 study, that's a study that's looking at induction therapy with bortezomib, lenalidomide, and dexamethasone, given in typical fashion as is currently standard of care, or bortezomib, lenalidomide, and dexamethasone, followed by a single Cilta-cel infusion and no maintenance therapy. In the CARTITUDE-6 study, patients received daratumumab, bortezomib, lenalidomide, and dexamethasone induction therapy, and they're randomized 1-to-1 to either autologous stem cell transplantation or Cilta-cel. There is 2 years of lenalidomide maintenance therapy applied after recovery from either transplant or Cilta-cel infusion, but it is a single Cilta-cel infusion. And CARTITUDE-6 will look to see whether we can replace transplant as part of the frontline paradigm going forward.